This is Dr. Matthews, and we're going to talk about some microbiological laboratory techniques. First is keeping in mind you've got to use aseptic technique when handling any microbes or for doing surgery or for anything where you don't want to contaminate your environment. And when you're transferring microbes from one environment to another, say from a patient to a petri dish, you have to use aseptic technique. No germs outside the ones you're needing to use. Transferring microbes usually have transfer loops and needles, and you sterilize the loop or the needle with a flame, and then let me draw this flame here. Here's our flame. And you're going to hold your loop over the flame. Here's my loop. Until this is burning red hot, and then take it off. Now this right here represents my petri dish, and I've still got it, and it doesn't have any ends on it. The correct procedure is not to take the lid off and set it aside, but to tilt it up. See all these sprinkles here? Those are germs falling. Germs fall with gravity just like anything else does. And so keeping things out of the top, always keeping your top covered is part of your aseptic technique. And what I like to do is heat my loop, and then if I have a clean petri dish with agar on it, I touch it to that and you can hear it sizzle like that. And you know it's cooled off. So then I will take it in to spread my bacteria out. Okay, that's with test tubes we have on the next page or two. Here's sterilization of the loop. This is not red yet, but it will get there. If you're going to use a test tube, you're going to need to flame the tip of it, and this is an art. You've got to run the flame over it. Of course, this is a still picture. If he just holds that, especially right there in the hotter part of the flame, the hottest part would be blue right up in here. But you just run it through it a few times. If you hold there, it's going to get hot enough that it's going to burn your hand and possibly break, and could throw your solution all over the floor. Okay, sterilization is destruction or removal of all microorganisms, and there are many different methods. Heat, radiation, filtration, and chemical methods are ones that are used. This is an autoclave. This is a very big autoclave. I have a much smaller one in my lab. It heats, and you need to use distilled water in these, because if you don't, you'll get mineral buildup and they won't last near as long. But you heat up your whatever you're trying to clean, glassware, instruments. They're used for surgical instruments, but you put them in here, you have water in there, you seal it down, and it heats it up at about 121 Celsius for 20 minutes, unless you've got a bunch of liquid stuff in it, and it may take a lot longer then. And it will kill endospores. Disinfection is just physical process and chemical process to kill the vegetative or growing microbes and active viruses. And this is for inanimate objects and some medical equipment, man-made objects, the kitchen table. We try to disinfect our skin, but we call it an antiseptic. We're not really going to use the same thing. You're not going to scrub your hands with Clorox, I hope. Sanitation is cleaning as many microbes off as you can. And soap, good old soap and water. Restaurants, dairies, breweries, other food industries, hopefully your hands got to be washed with soap. Now the thing about soap is it's got two ends. One of the ends is going to be I'll draw blue. This is hydrophilic. It likes water, and water will attach to this end. The other is hydrophobic, and I can't find the hydrophobic end, and grease attaches to that. And so you get your stuff all over this, you get your soap all over your plate, your restaurant, your instruments, whatever you're trying to clean, and you will have the water will rinse away the germs. Without soap, just try rinsing a greasy dish in the sink and not use soap. It doesn't work. Same thing with washing your hands. If you don't wash them with soap and water, pay attention to your fingernails and really get them clean, then you have not washed your hands and you're putting your patients at risk, yourself at risk. Degermination is removing as many microbes off the skin as you can. Scrubbing, you can immerse in chemicals or both. It's come in vogue in some places to just use chemical disinfection. Basically, there's a pump thing on the wall, and you just scrub your hands with it like a hand sanitizer type thing before surgery. I don't do that. I still wash my hands and scrub them thoroughly before surgery. I hope everyone who ever works on me does, but I'm usually asleep. Okay, when you're going to grow microbes, you have what's called the 5 I's. Inoculation. You're going to take your microbe, put it in your media that you're going to grow it in. Incubation, put it in an incubator. Isolation, separate out individual species. Inspection, look at them under the microscope. And then identification, based on morphology and other things. Types of media, we have liquid media. We call those broths. Semisolid media, gelatins. They are sort of clot-like at room temperature. They'll get solid at cold temperatures, and they can be used to determine motility of bacteria. Solid media, agar is a good example because it is solid at room temperature or refrigerated temperature. You liquefy it at boiling temperature, and then you can pour it in a dish and let it cool and grow your stuff on it. It's really handy because just using a gelatin, if you try to grow bacteria on a gelatin, it will liquefy at room temperature. It certainly liquefies in the incubator. Okay, media. This is what you're growing your stuff in, your bacteriums. They may be synthetic, and if they are, they're going to have some pure organic and inorganic, but it's a specific formula, and they're reproducible and standardized, so each bottle you get is identical to each other bottle. Others are not quite that way. Some are much more complex, and each is not exactly the same. If they have animal tissues, plant, yeast, ground up cells, tissue secretions, they're not going to be exactly the same. There's a lot of general purpose media that for microbes that are not hard to grow. And Tryptic Soy Agar is what's pictured here. Nutrient agar is another. The agar is going to be maybe 2 millimeters, 3 millimeters thick here, and these little specks are your bacteria growing. Enriched media, you may start off with just regular agar and then you add extra stuff. Like if you want to look for hemolytic strep, blood agar is just fantastic. Sheep blood or horse blood are preferred. When I worked at Fort Valley State, we had an old horse. If you'd go out there with a treat, you didn't even have to put a halter on, and she'd donate blood just gladly to make blood agar with. Now, if you're ever looking around in the refrigerator in the lab and you see something that says chocolate agar, don't eat it. Chocolate agar is blood agar that's been heated until it turns dark, so it's basically burnt blood. But if you have, say this is your petri dish, and you have hemolytic strep, this is our strep organism, then the blood around it is going to be gone. That kind of looks like an eyeball. Okay, selective media may inhibit a certain microbe. Maybe they inhibit gram-positives or gram-negatives. Some media are made to grow fungi on, so they have antibiotics in them. But these are for specific samples when you're looking for a specific thing. Differential media show differences, and they may have If you have a particular organism, it may be pink, and another organism may be blue. And I wish that your book had pictures of those because they're pretty cool, and I should have looked them up, but you can look them up. You can't grow everything in cell culture. Why can't you grow viruses in a culture plate and put it in the incubator? Because viruses are not living organisms, remember? They have to have a cell to grow in, so you can grow them in cell culture. Duck embryos are something that are often used to grow things like vaccines in. I know rabies vaccine. They came up with a human cell culture that they were growing rabies vaccine in, and they're more using the duck embryo now just because of the expense. Believe it or not, it's cheaper to get a duck to lay an egg than it is to have a tissue culture donated from somebody. Okay, inoculation. This is your putting your bacteria to produce a pure culture. And you have to have a medium for it and a sterile inoculating loop or needle and sterile cotton swabs to take it from the patient. So you might, well, I'll just show you how you make a street plate right here. Okay, we have our sterile cotton swab. Let's see what we want to use, a highlighter maybe. And we're going to initially take and make streaks with that swab. We've already dipped it in the abscess on Mr. Brown's back. These should be straight lines, but this computer program won't make straight lines. So you're going to do that. And then you're going to take your sterile loop, heat it up, put it right over here where it'll be cooled off, and you're going to run it through Mr. Brown's bacteria. Blunk, blunk, like that. Okay, and just to show you that we're making something different, I'll get a different color. So here, and we're going to take this we've heated this up. Right over here, you know there's no bacteria. You may not see it very well. And it just cools it off. And then you run it through here. What we're doing is we're getting thinner and thinner and thinner. Now let's make the last ones. When we already used blue, we'll use purple. So we heat this up, and then you're going to run it through here, stopping before you get to the initial culture. I forgot to sizzle it first. Got to cool it off or you'll kill the bacteria. And once you get this grown, you will have thinner and thinner. This will be thick, this will be less thick, thinner. And here you should be able to spread this out to individual bacterium. And by doing so, you can then take a sterile loop and pick off an individual culture. Then you can respread that on another plate if you want to, just to be sure you've got what you need. Or you can go ahead and look at it, stain it, and look at it. Incubation, you're going to put it in your incubator. Now some cultures are grown at room temperature. I know a lot of fungal cultures are grown just at normal room temperature. Most things you're going to grow at 37 Celsius, which is human body temperature. Then you're going to isolate them, and hopefully you're going to have colonies, little bumps of organisms that grow. That's a bump of an organism. It's a colony of organisms that grew from a single cell and therefore contain a single species. And you can get pure cultures from several ways. Your streak plate, which I already mentioned, and this is what it looked like, the one I streaked out for you in Imaginary World. Here it is, and here you have just individual colonies, and every bacterium in this should be the same. Now if you had a pink one and a yellow one and a green one, those are going to be different. And then you're going to have to restreak them and try it again. Okay, a pour plate, you've added your inoculum to warm, not hot, but warm liquid agar and poured it in the plate, and it solidifies, and you've got bacteria all through here. And you can see that you have differences. There's some heavy growth. This looks very different. This looks very different. And you'd have to look at these microscopically to know what you've got. Spread plate, spreading this all over. This is really good when you're going to do culture and sensitivity to put your little discs on. We had the discs of disinfectant, I think, on your last show, but this one we don't have any discs to put on it. But there we go. Hopefully you've got some on another slide. Oh, we do in lab. Okay, and here's how your spread plate grew. Now, fixation and staining. You can look at living organisms, but they usually work out better if you look at them dead. And you take a smear. Here's your slide. And again, in lab, I will have some pulled up slides of people actually doing these things. And you're going to take a very small amount and put it right in the middle and smear it out. Some people add a drop of water there to spread it thinner, and then you need to allow it to dry. Then heat fix it. Now when you heat fix, you're not going to just hold the glass slide here and hold it and hold it and hold it and hold it until it's so hot that you break it. You're going to wave it over it like this. It's almost an art. If you get burned, you did it wrong. If your organisms are still alive, you did it wrong. So you're going to heat fix it. Then you're going to stain it. Some stains are negative stain. India ink is one. It's black in the background. These are really good for yeast capsules, spirochetes, bacteria with capsules. So you have a dark background, and you don't have to even fixate these. So here is a Klebsiella pneumoniae. The background is dark. These are your organisms. Simple stain, you're just using one stain. Methylene blue, crystal violet, safranin are some of the ones that you could use. And here's one they use crystal violet on Bacillus cereus. Differential stains take more than one stain. The gram stain is my main go-to in differentiating things because it helps me know what antibiotic to use. It's a 4-step process. Now, the gram-positive stains a purplish color because of the peptidoglycan soaks up that purple. The gram-negative has a different type of outside cell wall that won't hold the purple once you put decolorizer on it. So gram-negative bacteria will be pink or red, and we might say, well, who cares? Well, different organisms, gram-positive organisms, respond to things like penicillins, cephalosporins, whereas gram-negatives may respond to streptomycin, gentamicin, and then there are some broad spectrum antibiotics. But I like to look at these, and it gives me an idea of what to treat with. Okay, and this is gram staining process. You have spread your thing out, you have heat fixed it, you put crystal violet on it for 1 minute. You've already let it dry too, 1 minute. Then you rinse it with tap water. Then you grams iodine for 1 minute, rinse with tap water. Then you decolorize with alcohol. It can be ethyl alcohol, it can be ethanol acetone, it can be isopropyl alcohol. Most of the little kits come with isopropyl or ethyl alcohol. And when the kit runs out, I usually just go ahead and buy isopropyl. Now, ethyl alcohol is the same alcohol that's in alcoholic beverages, but if you go into the pharmacy and buy ethyl alcohol, it will be denatured. And if you drink it, it will make you barf repeatedly. That's because they denature it. They put this toxin in it. It doesn't kill you. It just makes you wish you were dead, so they don't have to pay the liquor tax on it. Okay, you rinse that with tap water, and this is a 5-second thing. So we've got 1 minute, 1 minute, 5 seconds. then you rinse. Then safranin is the red stain, or pink. You let it sit 1 minute, rinse. It says blot dry. I'm very careful about that. I often just stand mine up on edge and let it dry, air dry. If you blot it, be sure that you blot and don't rub. If you rub it, you're going to rub all your organisms off, and then you've messed up. Oh, and if you run your water hard, you're going to wash your organisms off. Okay, acid fast stain, and I have a slideshow somewhere in the lab class that shows how you do an acid fast stain. And acid fast bacteria remain red, non-acid fast are blue. So it's exactly the opposite of what the gram staining does. But if you know it's acid fast and gram positive, then you can narrow down what the organism is. This is an acid fast organism. Now, there are some special stains, negative stains to look for certain kind of capsules. There are endospore stains to look for endospores. Flagella stains to actually look for the flagella, the bacterial flagella. Now, Wright stain is generally the preferred stain for staining human tissues with, unless you're looking for something like malaria in a blood smear. Then you'd use a Giemsman stain. But this is a pretty simple stain to do, very easy. It's just, one stain, 30 seconds, rinse. Another stain, 30 seconds, rinse. And you get the pinks and purples. These are red blood cells stained. These are white blood cells. They're purple. How can they be white blood cells? Because we stained them. And these would be clear if we didn't stain them. Now, if you notice this, see how this is not taking stain there? If you remember from anatomy, the shape of a blood cell, they're sort of a concave. This part didn't take much stain. They're not like a balloon. They're like a little disc that's been smooshed in. Or maybe like a donut someone was going to punch the hole out on and gave up. Okay, this was probably a monocyte. They're hard to look at and tell from these angles. This looks like it's probably a neutrophil because you see it's got the elongated. This would be an immature neutrophil though. That's probably a platelet. Okay, now to the next slide. Identification techniques. You look at the morphology, and we looked at all those shapes and also physiological characteristics and chemical characteristics. Some of these which we've already done when we've done our staining. This was in another chapter, the bacilli. The biggest thing, the bacilli are the rods, and you can call them rods, but they're still bacilli. The cocci are round, and if you have a lot of bacilli and you see a few round ones, they could be sitting on end where you're looking down at this angle. Spirilli, vibrios, spirochetes, coccobacilli, it might be something like that. Okay, culture plates. We have nutrient agar. That's a typical steady go-to lab plate. Tryptic Soy Agar, phenyl alcohol agar. And these are selective for gram-positive bacteria. These are both general purpose. Blood agar, the biggest reason you would use blood agar and chocolate agar, looking for things that hemolyze, looking for organisms that hemolyze blood. And these chocolate agar can grow some respiratory bacteria that won't grow otherwise. Thayer-Martin agar is a type of chocolate agar that's for Neisseria. Look at that species name, gonorrhea. Okay, McConkey's agar, gram-negative versus gram-positive bacteria, very commonly used in urine cultures. Xylose lysine deoxy chocolate agar for culture of stool samples. inhibits gram-positives, facilitates gram-negatives. And since most of your stool samples and your stool pathogens are going to be gram-negative, then that would be a good reason to use that. Mannitol salt is going to differentiate organisms that ferment mannitol. Now, gelatin stabs. Let's look at the picture. I think would be the better thing. There we go. There's a stab. Now, before he did this, I'm saying he, guessing. It could be a she. She doesn't have fancy fingernails, but then neither do I. So you would have to take and this stab needle. It's not a loop, it's just a needle. You would have to first sterilize it over your fire, and then you dip it in your culture, your sample. Maybe you've taken a swab of somebody's abscess or a disease somewhere, and you can actually take this and put it on the end of this and then holding it. Notice the angle, because which way do germs fall? Down. You don't want germs coming out of the sky into here, so you just hold it sideways and just stab it in and pull it out, and then keep it sideways until you get your lid on, and then you incubate that. Some of the fungal culture kits you buy come like this, and most fungal cultures where you're looking for skin cultures, ringworm, mycorrhizal fungal cultures are going to take several weeks to grow, like your patient's well or dead time the culture comes back. So I usually treat looking on the morphology of the culture and still do the culture if I can. I mean, looking at the morphology of the swab. Okay, growth patterns in broths. Here we go. You could get all your bacteria. It can grow at the top. They may all go settle to the bottom, or they may be throughout this. And different organisms like to grow differently, so this could help you differentiate your organisms. To actually get down to the final, we know what this is, you're going to have to do a genetic analysis. Strep, Streptococcus pyogenes may be nothingness. It may be making, it's just a normal bacteria growing in your throat you're used to. It may be causing strep throat, meningitis. It could be causing flesh-eating disease, and it all looks the same under the microscope. You're going to have to get down to looking at your DNA or RNA, and these are going to take a little bit to do to actually sequence the DNA, and hopefully you'll have a pretty good idea pretty quickly, because if you don't, and it's flesh-eating bacteria, your patient will be dead in time the culture comes back. This is insurance companies want you to wait to treat sometimes until your culture is back. But I look at it like, if the patient's dying today, we'll go ahead and treat him, see what the culture says later. Okay, that's all for now.